In a previous homework, you learned about the four major macromolecules: carbohydrates, lipids, proteins, and nucleic acids. The purpose of the digestive system is to break these macromolecules down into their smaller subunits called monomers so that they can cross the cell membrane and be used by cells for energy, growth, and repair. There are four stages to food processing. The first is ingestion, or taking food or water inside the body. The second is digestion. There are two different types of digestion. The first is mechanical, or the physical breakdown of food into smaller pieces. The second type is chemical digestion. This often involves enzymes to digest food into simpler molecules. Then these molecules are absorbed. They cross the cell membrane and enter cells. The last stage is elimination, where undigested material is removed from the body. Digestion begins in your mouth with mechanical and chemical digestion. An example of mechanical digestion would be using your teeth and tongue to chew your food. Enzymes in your saliva would be an example of chemical digestion that helps break down food. From your mouth, your food travels through your pharynx and down your esophagus. Peristalsis, or the wave-like contractions of muscle, help move food down the esophagus and into your stomach. Your stomach is a highly acidic muscular sac. Here, there is hydrochloric acid and more enzymes to assist in the breakdown of food. When food leaves the stomach, this acidic liquid is called chyme. Chyme moves into the small intestine, where nutrients are absorbed, then into the large intestine. Water, salt, and some vitamins are absorbed here. Organs that are not part of the alimentary canal, which means that food does not travel through them, but still help with digestion, are called accessory organs. Here we have the liver, gallbladder, and pancreas. All assist in the digestion of food. The liver secretes bile, which is stored in the gallbladder and then released into the upper small intestine to help break down fats. The pancreas secretes pancreatic juice that helps neutralize the acidic chyme coming from the stomach. The structure of the small intestine maximizes surface area. The small intestine itself is very long and highly folded. Each fold has finger-like projections called villi that maximize surface area. Inside one of these projections, we have capillaries. Food travels from the inside of the small intestine, crosses the cell membrane, and enters our bloodstream. This is absorption. We mentioned enzymes when we were talking about chemical digestion. An enzyme is a complex protein that lowers the activation energy of a chemical reaction. We refer to enzymes as biological catalysts. They help speed up reactions, but are not used up during the chemical reaction. This means that they can help the reaction occur over and over again. Enzymes can be impacted by certain environmental factors, like temperature or pH. Here we see two examples of reactions. One is energy absorbing, in that its products will contain more energy than the reactants did to start. The second is energy releasing, so the reactants produce energy when the reaction occurs. Most individuals think of energy releasing actions when they think of enzymes. However, enzymes can be used to both build larger molecules, synthesize larger molecules, or to break down or decompose larger molecules. Enzymes make it more likely for both of these reactions to take place than they would without an enzyme present. A certain amount of energy needs to be invested so that a chemical reaction will start. In the graph above, we see two different chemical reactions. The gray line is without an enzyme, and the black line is with an enzyme. With an enzyme present, the activation energy is significantly lower than without the enzyme. This image shows an enzyme-catalyzed reaction, or a chemical reaction that is taking place with the help of an enzyme. In this type of reaction, we refer to the reactants as substrate. You will notice that the substrate matches up to the enzyme in this image. That is meant to show that substrates are specific to their enzymes. Enzymes only bind to certain substrates. I want to thank you all for joining me for this overview of enzymes in the digestive system. Please let me know if you have any questions.